Okay, so for the first part of today's lecture, I'm going to be using the three-dimensional structures of myoglobin and hemoglobin to demonstrate the similarities and the differences between these two proteins. And I'm getting these three-dimensional structures from an online database. I showed you a couple of databases last week, and the one we're looking at today is this one here, the RCSB PDB, that is the Protein Data Bank. And this is an international structural biology knowledge base. And when a researcher solves the three-dimensional structure of a protein, generally using X-ray crystallography, they will deposit the coordinates of every atom in that structure into this database. So in this database, there are tens of thousands of protein structures, everything from cytochrome C up to full virus particles. And a couple of things I want to show you on here. So one thing is this thing here, the PDB 101. And this is kind of cool because it has this molecule of the month feature. So this is the entry for hemoglobin as molecule of the month. So it gives you all kinds of background information about this molecule. And it also gives links to, so this thing here says 2-HCO. So this would be a structure, or one structure of hemoglobin. We have other structures of hemoglobin here because hemoglobin has been crystallized with various ligands. So it's been crystallized in the presence and the absence of oxygen. And this little GIF here is showing you the differences, how the structure of hemoglobin changes as it binds and releases oxygen. And this is going to become important when we're talking about how it is that hemoglobin can pick up oxygen in the lungs and then deposit it in the peripheral tissues. We have stuff about hemoglobin mutations we're also going to talk about in more detail. So this is a really nice, the PDB is a really nice resource, not just for the actual structures for themselves, but also for detailed information about these structures. Now the two molecules that we're going to compare today are myoglobin and hemoglobin. So let's start with myoglobin. And this is just one of the structures of myoglobin that's been deposited here. This particular one is a horse heart myoglobin. And there's all kinds of information. These tabs here provide you with tools to look at the sequence, to search for proteins of similar sequences, to look for structural similarity, and then also a lot of literature about the protein itself. Now what we're going to do is I'm going to open up this structure in this thing here, Protein Workshop. So on the left hand side here we have this 3D standalone viewer. And we're going to start by looking at the structure of myoglobin in some detail. So this is a single molecule of myoglobin. And I'm actually going to go in, I'm going to take those out. Make things a little bit more clear right now. So what we have here are 8 alpha helices bundled together to form this compact structure. And you can see there's a cleft in the middle, and this contains the heme group. So if we take out the protein backbone and we just look at the heme group by itself, so you see what we have here. So we have this hydrocarbon ring with 4 nitrogens here. And these nitrogens coordinate, that is, they hold in an ionic interaction, they hold this ion atom in the center there. Let's pull the protein chain back. And this heme group is nestled in a pocket in the protein. Now we're just looking at the backbone of the protein now. So we're looking at it from, so here is the N terminus, and you'll see, so what we're doing is we're following the backbone. So the peptide backbone, so we're going NCC, NCC, NCC along the backbone. So we're not seeing any of the side chains. So we can see we have these alpha helices bundled together. And if we look at the side chains, we'll see that this is actually a very compact globular structure. So let's make those side chains visible. Like that. So this is making them visible as a space-filling structure. So you can see that we actually have a very compact mass of density here. We can change the style of that. like that. So this is just a ball and stick representation. This makes it a little bit easier to see what the amino acid residues actually are. So I can see an aspartate over there, and then what else? So we're going to see principally these. Okay, there's a lysine on the surface there, as you'd expect. There's a glutamic acid. As you'd expect, we have more of the polar amino acid residues on the surface. And then in this cleft here, we actually have more non-polar amino acid residues, which help to bind this heme group there. So let's make those go away. Like that. Now there are two particular amino acid residues I want to highlight in this structure. So if we open up the chain for myoglobin and we scroll down, and I want to bring your attention to two histidine residues. So a histidine residue at position 64, so that's that one here, and then another histidine residue at position 93. And this is the nice thing about looking at this in 3 dimensions, because you can really see what's happening here. So this one here at 93, this is what we call the proximal histidine, that it's in close proximity to the iron atom in the heme group. Then down here we have what's called the distal histidine. So you see we have this space here. It's a bit further away from that iron group. So if you're going to guess which side the oxygen binds on, you'd say, you know, I think the oxygen binds here between this histidine and this iron atom, and that is in fact the case. So things we need to notice. So we have our heme group with the iron atom coordinate in the center. We have 2 histidines pointing directly, forming a 5th and 6th coordination site, and that myoglobin is a monomer comprised of 8 alpha helices. So let's have a look at the protein that's responsible for picking up oxygen and transferring it to myoglobin in the tissues, and that would be hemoglobin. So here is a structure of hemoglobin. Let's make this a bit bigger so you can really see it. And this looks a bit more complicated. You can see right off that this is a more complex molecule than myoglobin. And in fact, what we have here this is a heterotetramer. It has 2 alpha chains and 2 beta chains. And I can show you those. Let's make those visibility ribbons. So if you make the A chains go away, like that, and then like that, you can see that in fact we have 4 chains. Now what I'm going to do, so this one that we're looking at here, this is actually again in the deoxy form. So you can see that we have a heme group, but with no oxygen bound. So I'm going to go to a different structure, and we're going to have a look at the oxyhemoglobin in a bit more detail. Let's just see if I can find that. Okay, here it is. So the previous structure I was showing you was hemoglobin with no oxygen bound. So what we're going to do now is going to look in a bit more detail at hemoglobin with oxygen bound. So again, this is 2 alphas and 2 betas. So the blue are the A chains, and the green are the 2 B chains. And you can see they're actually separate chains. So you can see there's a gap between the end of this one here and the beginning of that one there. And that we have these 4 heme groups. And I'm just going to zoom in. I'm going to take the A chains out to make it a bit more obvious what's going on. And then I'm going to zoom in on just one of these heme groups here. And what I've also highlighted are the equivalent histidines in the in this hemoglobin. So we have a proximal histidine. So this is histidine 92 in this sequence, and then we have a distal histidine, which is histidine 63. And nestling between the two, or nestling between the distal histidine and the iron atom, there you can see that molecule of oxygen is bound in there. So the questions that we're going to answer in today's lecture, let's just get back those chains back. Okay. So the questions we're going to answer in this lecture is really to use this structure and the biochemistry of this structure to help us to understand how it is that this molecule performs its function of picking up oxygen in the lungs and offloading that oxygen in the peripheral tissues, and how myoglobin performs its function of taking up that oxygen that is deposited by hemoglobin. And the key to understanding how hemoglobin can perform its function is understanding why it's a tetramer and why myoglobin is a monomer. So we're going to go into that in more detail in the actual lecture. Okay, so let's talk in more detail about the structure and function of these two proteins. So hemoglobin and myoglobin, they have some similarities. They both bind oxygen at ion-containing heme groups. They share sequence similarities, but they definitely share very, very similar tertiary structures. However, hemoglobin is a tetramer, whereas myoglobin is a monomer. One of the things we're going to talk about, and I will explain this in a moment, is that the binding of oxygen to hemoglobin is cooperative, and it's affected by a number of regulators, by the pH of the environment and the binding of a molecule called bis-phosphoglycerate. The binding of oxygen to myoglobin, in contrast, does not display cooperativity. So we're going to explore the differences in structure and oxygen binding at the molecular level, and we're going to relate them to the physiological roles of these two proteins. So, as I said, they have many similarities. So they're both globular proteins comprised of alpha helices. They have a hydrophobic cleft binding the ion-containing heme group. Now, hemoglobin's function is to transport oxygen to peripheral tissues, and it also transports some carbon dioxide back to the lungs. When hemoglobin binds allosteric activators, it changes in its structure, and this regulates the oxygen binding and release. And incidentally, hemoglobin, a mutant form of hemoglobin, was the first characterized inherited molecular disease. Myoglobin, also globular protein, alpha helices, also has a heme group, and this is the primary oxygen carrier in muscle tissues, and it does not display allosteric binding of oxygen, which I'm going to explain as we go along. So both of them contain this thing called the globin fold. It usually consists of 8 helices, and they're packed in 2 antiparallel layers with a 50-degree angle between them. It's common to hemoglobin and myoglobin, and also a wide range of other proteins with similar function. Neuroglobulin would be an example. So we already saw that both of them have this structure with the histidine residues pointing at the iron atom. And what we're seeing in the middle here, so this is a structural comparison that I did of a myoglobin monomer and also one of the beta hemoglobin monomers. So you can actually overlay the two. So you see the myoglobin is in blue and the hemoglobin chain is in this tan color. And you can see that there's a lot of similarity between the two. And one of the questions we're going to answer now is, why does hemoglobin have to be a tetramer? So both myoglobin and hemoglobin, they bind oxygen at the heme groups. So we have this thing here, it's called the protoporphyrin ring, with a central iron atom, and it's in the Fe2+, in the ferrous state. So it's coordinated to 4 nitrogens in the center of this protoporphyrin. And iron can actually form 2 additional bonds above and below the plane of this ring, and they're called the 5th and 6th coordination sites. Now, the 5th coordination site is occupied by an imidazole ring. So this is the histidine we saw called the proximal histidine, and we can see it here. So here's the proximal histidine coordinating to iron. And the 6th coordination site, that's between the distal histidine and the iron, binds oxygen. So an interesting thing to note, and this is key to the function of hemoglobin, is that when oxygen binds to hemoglobin, the iron moves into the plane of the protoporphyrin ring. So you see in this one here, the iron is sitting a little bit below the plane of the protoporphyrin ring, which is also slightly kinked. When oxygen comes in, it pulls the iron up into the plane of the protoporphyrin ring. And interesting, what happens, this actually tugs on this histidine residue here, and this is one of the things that causes a conformational change in the protein when oxygen binds. Now, what we're looking at here is a comparison between how much oxygen is bound by myoglobin or hemoglobin at any given oxygen concentration. So this PO2, and the units for this are torr, this is what we also call partial pressure, is really a measure of the saturation of the solution surrounding the proteins with oxygen. So we can go from 0 to 100% saturation of the solution with oxygen. And what we're looking at here, so this then is a measure of how saturated the myoglobin or the hemoglobin is with oxygen at any given partial pressure of oxygen. So let's look at the myoglobin first. So as we increase the oxygen concentration, we can see that the myoglobin binds oxygen. So it becomes saturated with oxygen. At 1, that would be complete saturation of all the myoglobin molecules with oxygen. So not surprisingly, the more oxygen there is available, the more the myoglobin molecules are saturated with oxygen. Now, interestingly, we compare that with the hemoglobin curve. So starting at these low oxygen concentrations down here, we'll see that whereas as we increase the oxygen concentration, the myoglobin rapidly loads up with oxygen, the hemoglobin does not. In fact, it doesn't really start loading up with oxygen until you get to a much higher oxygen concentration, and then its rate of loading, or the degree to which it saturates, is much slower with the increase in oxygen concentration. And this is an example of something that we call allosteric cooperativity. So what happens is we have these 4 subunits of hemoglobin, each of which can bind an oxygen. When no oxygen is bound whatsoever, the hemoglobin is kind of in an off state that doesn't really want to bind oxygen. But once one oxygen is bound at one subunit, this causes a conformational change. It's communicated to the other subunits. This causes changes in the overall structure of the protein, which promotes oxygen binding. So we start off with no oxygen bound. As the first atoms of oxygen bind, this causes a conformational change, which makes subsequent oxygen binding easier. So let's have a look at models for cooperativity. So many of these multimeric, that is multi-subunit proteins, and we're going to see this again and again in enzymes as well, they exhibit this behavior called cooperativity. And there are 2 main models for cooperativity. Now, all of the models postulate that the enzyme or the receptor, like hemoglobin, it has binding sites that exist in one of 2 states. We call it the T state and the R state, where T stands for tense and R stands for relaxed. And the T state has low binding affinity for the ligand, the R state has high binding affinity. And we have 2 major models, the Mono-Wyman-Changeur model, and this says that binding of a ligand to one site results in a global conformational change in all of the sites. So here is our example. So here we have, so this, for example, could be our hemoglobin tetramer, and in the absence of oxygen, all of these sites are in the T state. However, once one oxygen atom becomes bound to one of these subunits, it causes a conformational change in the entire protein, so it's communicated to the other 3 subunits, so they go from the T to the R state. There's a second model called the KNF, or Koshland-Methian-Filmer model, and this is what's called a sequential model. And it says here the binding of ligand to one site induces a conformational change in that site, and that this then propagates a conformational change in adjacent subunits. So you have instead of having a global change where they all go from T to R, you have this one site goes from T to R, and this is partially communicated to the other sites. So that then as a second one binds, this would then communicate, and so on and so on. So you have a sequential model of binding and gradual conversion from R to T, from T to R state. Now we can see we have X-ray crystal structures for both deoxy and oxyhemoglobin, and these show conformational changes on oxygen binding at both the subunit interfaces and the heme binding pocket. So what we mean by the subunit interfaces are the interfaces between these 4 protein chains. So where the 4 protein chains nuzzle up against each other, we call those the interfaces, but also in the heme binding pocket itself. So the transition from deoxyhemoglobin, the T state, to oxyhemoglobin in the R state occurs upon oxygen binding. So remember, the iron atom, as oxygen binds, the iron atom moves into the plane of the heme. The proximal histidine, which is a component of an alpha helix, moves with the iron. So what we're looking at, if we look at this structure here, let's see if we can get there. Okay, there's the mouse. So when oxygen binds, you'll see that the histidine, that the heme group shifts, and this thing that we're looking down at here in yellow, this is the proximal histidine. And as oxygen binds, there's a tug on that proximal histidine, which causes a conformational change. You see this tan-colored alpha helix here, and this histidine is part of this alpha helix. It actually causes a tug on this, which helps to induce a conformational change in the protein. So oxygen binding induces a conformational change, which transforms this subunit from the T to the R state, from the tense to the relaxed state, and this conformational change is also communicated to the other subunits, the other 3 chains in the hemoglobin tetramer. Now, the resulting structural change, what happens is it's communicated to the other subunits so that the 2 alpha-beta dimers rotate with respect to one another, resulting in the formation of the R state. And as I say, we have crystal structures for both deoxy, that is T state, and oxyhemoglobin, that is R state, and they show many conformational changes on oxygen binding, as I say, both at the subunit interfaces and the heme binding pocket. So this is just one of the changes that happen at the subunit interfaces. So here we have, what we're looking at here are 2 alpha subunits. So this is in the T state with no oxygen, and you can see we have an intermolecular ionic interaction between lysine at position 127 and arginine at position 141. So this is on one chain. And then on the other alpha chain, we have a same ionic interaction, lysine 127, arginine 141. Now what happens when oxygen binds is that we actually now get an ionic interaction between arginine 141 and arginine 141. So this is an intermolecular interaction. So this is intramolecular within one molecule. This is intermolecular between 2 molecules. So you see we've had this big shift in the conformation, in the interaction between the amino acids of these 2 subunits. So this cooperativity actually leads to efficient delivery of oxygen by hemoglobin. So when we look at this saturation and desaturation curve, so if myoglobin were the oxygen carrier in blood, only 7% of its oxygen would be unloaded in the tissues. So what do we mean by that? So let's compare. So the oxygen concentration in the lungs is about 100 torr. The oxygen concentration in the tissues is about 1/5 of that at 20 torr. So if myoglobin were the oxygen carrier, so in the lungs it would be 100% saturated, and then in the tissues at 20 torr it would only lose 7% of its oxygen. Hemoglobin, in contrast, in the lungs it's pretty much 100% saturated, so we have 1 is the fractional saturation, and then when hemoglobin goes to the 20 torr oxygen environment in the tissues, it loses 66% of its oxygen. So what happens then is once oxyhemoglobin reaches the tissues, it unloads oxygen cooperatively. So once one oxygen unbinds from one site, it's going to tend to that site will change into the T state. It's going to promote the T state conformation in the other subunits, leading to more unloading of oxygen. So hemoglobin, because it can undergo this R to T state transition, can unload oxygen very effectively when it's in this low oxygen environment. Now, oxygen binding is not the only factor in the T to R transition. There are other things that regulate whether hemoglobin is the T state, that is the low oxygen binding state, or the R state, the high oxygen binding state. One of these is a molecule called 2,3-bisphosphoglycerate. This binds to hemoglobin and it stabilizes the T state. Also, if you change the pH of the environment, if you reduce the pH, you actually promote formation of the T state. So if hemoglobin goes into an environment with a lower pH, it's actually going to unload its oxygen. And the other thing is reaction of carbon dioxide with terminal amino groups on hemoglobin also causes a conformational change which promotes formation of the T state. So remember, in this T to R transition, when you go from the R state to the T state, what you're doing is you're promoting unloading of oxygen. So 2,3-bisphosphoglycerate, reduction in pH, or the presence of carbon dioxide will promote the unloading of oxygen from the hemoglobin molecule. So as I already said, this 2,3-bisphosphoglycerate stabilizes the T state of hemoglobin. So we look at the binding curve. So this one here, this is hemoglobin, as it would be in red cells with 2,3-bisphosphoglycerate. If you purify hemoglobin, so you just have pure hemoglobin extracted from blood cells, it doesn't have any bisphosphoglycerate. And when you look at its binding curve, it actually looks a bit more like myoglobin. In fact, it looks a lot more like myoglobin. It doesn't have this same allosteric cooperative loading and unloading, and so that if you take this pure hemoglobin from the high oxygen concentration in the lungs to the low oxygen concentration in the tissues, it only loses 8% of its oxygen. Whereas hemoglobin with 2,3-bisphosphoglycerate binds acts in this classically cooperative state, so it's fully saturated in the lungs, and then as it goes to the tissues with this low oxygen concentration, it unloads 66% of its oxygen. And the bisphosphoglycerate actually binds in this central cavity. So these two yellow ones here, this is the two beta subunits, the pink ones the alpha subunits, and it binds in this cavity between them. So you can see it's stabilized by these ionic interactions with charged amino acids, and when it binds there, it promotes the formation of the T state. Now, another thing we want to talk about is the effect of pH on the affinity of hemoglobin for oxygen, and this is called the Bohr effect. So if you lower the pH, this causes key residues which normally form ion pairs to protonate. This results in a conformational change which reduces hemoglobin's affinity for oxygen. That is, you get a conformational change, and the hemoglobin goes from the R state to the T state, and it unloads its oxygen. So if we compare the binding of oxygen to hemoglobin at pH 7.4, so that's this pink line, and pH 7.2, you'll see that at pH 7.4 you can get, you have the shift in the curve over to the left side, so that the hemoglobin is more saturated with oxygen, but also loses less oxygen when it goes to the oxygen concentration in the tissues. At a pH of 7.2, the hemoglobin can offload 77% of its oxygen in the tissues, because the pH of 7.2 promotes the T state, that is the state which will tend to offload hemoglobin. And it turns out that the pH where the hemoglobin is loaded with oxygen, that is in the lungs, is 7.4. So under these conditions, so at high oxygen and at a pH 7.4, hemoglobin loads up with oxygen. Now in the muscle, the pH is a little bit lower. It's 7.2 now instead of 7.4. So you get this conformational change in the hemoglobin as the hemoglobin is transported out to the tissues. So it goes from the R state to the T state, and it unloads its oxygen. So what's the actual chemical basis of the Bohr effect? So in deoxyhemoglobin, there are 3 amino acid residues which form 2 salt bridges that stabilize the T state, this T quaternary structure. And the formation of one of the salt bridges depends on the presence of an added proton on histidine in the beta subunit at position 146. So here's a histidine, and this is a protonated histidine. So to form this salt bridge, it has to be protonated here. Now, the proximity of this negative charge on aspartate beta 94 favors the protonation of this histidine. So this aspartate group here is favoring the protonation. Now, you should notice that the salt bridge between histidine beta 146 and aspartate beta 94 is stabilized by a hydrogen bond. So that's this green dashed line here between these two residues. So the presence of CO2 decreases the affinity of oxygen beyond the effect of decreased pH. So we have these additive effects here. So let's have a look at these curves here. So this is the pink one here, or this reddish one here, is pH 7.4 with no carbon dioxide. So remember, this is a T-state style hemoglobin. Or I'm sorry, rather an R-state style hemoglobin. At pH 7.2, with no carbon dioxide present, you get the shift in the curve here to the right, so it's going more into the T state. And at pH 7.2, with the addition of carbon dioxide, it's even more pronounced. This allosteric effect is even more pronounced. So at a pH of 7.2, in the presence of carbon dioxide, hemoglobin goes into a conformation which promotes unloading of oxygen. So the presence of carbon, as I said, the presence of carbon dioxide decreases the affinity of hemoglobin beyond the effect of the decreased pH. So it's not just affecting the formation of the salt bridge by protonating the histidine. And the second reason for why carbon dioxide decreases the affinity is that the carbon dioxide reacts with terminal amino groups to form negatively charged carbamate groups. So here is an amino group on the N terminus of hemoglobin. It reacts with carbon dioxide to form a carbamate that then can form salt bridges that stabilize the T state. Now, interestingly, the hemoglobin A and B chains are examples of divergent evolution. So their function was maintained even though the sequence was not. So remember, I showed you these histidine residues. So here's an alignment of hemoglobin A, hemoglobin B, and myoglobin. And you can see that there are differences between the amino acid sequences. Not surprisingly, there's more similarity between hemoglobin A and hemoglobin B, but there are still significant differences. And myoglobin is very, very different in sequence from hemoglobin. The things you should notice is that in all cases, these conserve histidine, so the proximal and distal histidines are conserved. Let's put all this together. So we've talked about a lot of different effects, so let's put it all together and talk about how this then promotes the physiological role of hemoglobin. So, allosteric cooperativity of binding of oxygen to hemoglobin is mediated by allosteric regulators. This allows hemoglobin to efficiently bind oxygen in the lungs and then, just as importantly, to offload that oxygen in the tissues. So let's start off in the pulmonary capillaries with this high oxygen concentration. So oxygen will bind to the hemoglobin in the capillaries, and this is going to promote the formation of the R state, leading to efficient loading of all the oxygen binding sites on the hemoglobin. So in this high oxygen concentration in the lungs, hemoglobin will go do the T to R transition and will load up with oxygen. Now, hemoglobin, however, also usually has 2,3-bisphosphoglycerate bound, and this will tend to stabilize the T state, especially at lower oxygen concentrations. The third thing we need to think about is reducing the pH promotes the formation of the T state. So if we go from pH 7.4 to pH 7.2, we're going to tend to unload oxygen. And reaction of carbon dioxide with terminal amino groups on the hemoglobin also promotes formation of the T state. So let's put all of this together. So here we are in a pulmonary capillary. So here we have a red blood cell passing through the lungs. So let's remember what the conditions are in the lungs. So the pH is 7.4, so a higher pH. The oxygen concentration is high. So under these conditions, hemoglobin will start to bind oxygen. So as a hemoglobin subunit binds oxygen, we're going to get this transition from the T to the R state. So that's going to open up the other binding sites on the hemoglobin, promoting oxygen loading. So under these conditions, hemoglobin is fully saturated. Now this hemoglobin molecule we then transported in a red blood cell out to the peripheral tissues. And in the peripheral tissues, the conditions are very different. So the pH is 7.2. The oxygen concentration is a fifth that of the lungs. We have a low oxygen concentration. So under these conditions, the hemoglobin is going to start offloading oxygen. So as an oxygen molecule comes off the hemoglobin, we're going to have the R to T transition. So this is going to promote further dissociation of oxygen. So oxygen molecules are going to come off. We're going to go from the R to the T state, and this is going to help offload our oxygen molecules. And this T state is further stabilized by the presence of the 2,3-bisphosphoglycerate, but also by the high concentration of carbon dioxide in the peripheral tissues. Now at this point, myoglobin, which doesn't have this T to R transition, it always has a high affinity for oxygen. It can bind this free oxygen. Now an interesting second case is in fetal red blood cells. So fetal red blood cells express a different type of hemoglobin. It's called alpha-2 gamma-2, where gamma-2, instead of the beta chains, this is the fetal hemoglobin state. Now the fetal hemoglobin doesn't bind 2,3-bisphosphoglycerate as well as maternal hemoglobin. So it makes fetal hemoglobin a bit more like myoglobin than it is like hemoglobin. So why does that need to be that way? This is because the only source of oxygen for the fetus is from the maternal blood supply. So at the placenta, the maternal red cells can give up oxygen, will unload oxygen and will pass it across to the fetal red cells. And this is made possible by this difference in the affinity of fetal hemoglobin compared to maternal hemoglobin. Now, hemoglobin was also the first molecular disease that was characterized. This was actually characterized by Linus Pauling back in 1949, and then further, the whole thing was nailed down completely in 1957. And he was the first one to show that sickle cell disease is a molecular disease caused by mutations in human hemoglobin. And what happens here is, so what Linus Pauling noticed was that sickle cell hemoglobin, so this mutant of hemoglobin which is responsible for the sickle cell trait, in a charged field, it had different charge properties. It was less negatively charged than the wild type hemoglobin. And the reason for this is that the sickle cell hemoglobin has a mutation from a glutamic acid at position 6 to a valine at position 6. Now, if you remember, at physiological pH, glutamic acid is negatively charged and valine is neutral. And what happens as a result of this mutation? So this mutant valine is actually exposed on the surface of the protein. So sickle cell hemoglobin is inherently less soluble than normal hemoglobin. That is, replacing that glutamic acid, which can form hydrogen bonds with a valine, makes this protein less soluble. Now, this is particularly a problem at low oxygen concentrations. And what happens is this hemoglobin S will tend to polymerize. So we can see this here. We tend to get the formation of these polymers of numerous hemoglobin subunits. Now, these polymers, these filaments of hemoglobin, will actually deform the red blood cells, causing this phenomenon called sickling. Now, these sickled erythrocytes are not efficient oxygen carriers, and they don't pass as easily through the fine capillaries in peripheral tissues, so they can actually block peripheral tissues, and will also lead to lower oxygen delivery to the peripheral tissues. Now, interestingly, even in heterozygous individuals, so individuals who only have one HBS gene, a full-blown sickle cell crisis can actually be precipitated by low oxygen conditions. Under normal circumstances, a heterozygous individual may suffer no ill effects whatsoever from the HBS, from having a single copy of the HBS gene, and also HBS hemoglobin in their erythrocytes. However, if you take these individuals to a low oxygen concentration, so for example, if you take them to altitude, you can actually precipitate a sickle cell crisis in individuals who were previously asymptomatic for sickle cell disease. And this actually happened, this was first noticed during the Second World War, when many US troops were taken to Colorado, to Fort Collins in Colorado for basic training. Now, Fort Collins is, I believe, at about 6,000 or 7,000 feet. So individuals have been taken from sea level up to Fort Collins, and some of them went into a full-blown sickle cell crisis as a result of being taken to altitude where there was a lower oxygen concentration. And what's actually happening here, so the interaction between this valine 6 on a beta chain of 1 hemoglobin and a hydrophobic patch formed by phenylalanine 85 and leucine 88 on a beta chain leads to this hemoglobin aggregation. So here we have the site of interaction. Here's the mutation at valine 6. Here's a little hydrophobic patch formed by phenylalanine and leucine. And especially with deoxyhemoglobin, so at low oxygen concentration, you get this aggregation and you get the formation of these fibers. And we can see here, so this is an electron micrograph inside a red blood cell, so we can see the fibers here. And there we can see a sickled red blood cell as a result of the formation of these fibers. Interestingly though, the sickle cell trait is remarkably persistent, despite the severe health problems, especially for homozygous individuals. So this means there must be some evolutionary advantage associated with this trait. And if we look at the distribution of malaria with the distribution of a sickle cell trait, so this is the distribution of malaria seen here in yellow, and this is the distribution of the sickle cell trait, and also similar traits like beta thalassemia, you can see that there's a significant overlap between the distribution of malaria and the distribution of the sickle cell trait. And it turns out malaria is actually a serious health problem. It kills 1.5 million people per year, and perhaps more of a problem is that it causes chronic debilitating illness in millions more, where people are essentially disabled and cannot be functioning active members of society. And individuals who have the sickle cell trait, that is where they're heterozygotes, they display an inherent resistance to malaria. So it turns out that where malaria is endemic, the trait is sufficiently advantageous to be maintained in the population. That is, malaria is such a problem that even with the problems associated with the sickle cell trait, it's still better to have the sickle cell trait than to be susceptible to malaria. And not surprisingly, the sickle cell trait is more common in certain ethnic groups where their origins in these areas. So 8 to 10% of African Americans have sickle cell trait. It's also common amongst Hispanics from Central and South America, South Asians, Caucasians from Southern Europe, and some Middle Eastern populations. Now, all newborns in the United States are now tested for sickle cell disease and sickle cell trait. So excuse me, I just have to stop to cough here. So the key points you need to take away from this lecture: hemoglobin is an allosteric protein. It displays cooperative binding. And you should be able to sketch the binding curves for hemoglobin and for myoglobin under different conditions. So myoglobin and hemoglobin bind oxygen at heme groups. You need to know about the distal histidine, which allows binding of oxygen to heme ion, and also reduces affinity of hemoglobin for carbon monoxide, and also helps to prevent oxidation of that heme ion from Fe2+ to Fe3+. The proximal histidine coordinates to the heme ion, Binding of oxygen to heme reduces the size of the heme ion, which then moves into the plane of the heme ring, causing movement of the proximal histidine, resulting in conformational change. You should also know that hemoglobin binds oxygen cooperatively. You should know which factors affect the T to R transition. So oxygen binding, the presence of carbon dioxide, bisphosphoglycerate, and the pH effects. You should be able to sketch the binding curves for hemoglobin with and without these allosteric effectors. You should also know that mutations in hemoglobin can result in disease and the molecular basis of sickle cell disease. In your textbooks, you should look particularly at, on page 162, question 18, the data interpretation problem. Okay, and that is all for this lecture. Thank you.